Thank you very much for giving me the opportunity to present and discuss our experiences using the OncoMine-Dx Express test assay with focus on lung cancer samples. In the first part of this presentation, I will focus on FFPE tissue samples, and the second part will focus on liquid biopsy. So these are my disclosures. This slide shows the intended use of the OncoMine-Dx Express test, which is a CE-IVD test. First, I start with a few information about our organization. So our organization was founded in the year 2019 in Stuttgart. The samples we receive are sent to us via the sending members, shareholders who are distributed throughout Southwest Germany. The molecular pathological analysis, including the preparation of findings, are carried out in our laboratory in Stuttgart. The reports then are in turn sent to the sending members and associates. From there, they are released and sent to the oncologists in the periphery. This ensures decentralized diagnosis and oncological orientation. And the main focus of our institution is molecular tumor diagnostics. This slide summarizes the main areas of investigation carried out in our institution. The scope of application of the OncoMine-Dx Express test assay in our institution mainly comprises the entities which are outlined here in red, and one of the largest areas of application for the OncoMine-Dx Express test in our lab are lung cancer samples, which are the focus of this presentation. So as I said, I will focus on lung cancer samples. And when analyzing tissue lung cancer samples, we often face several challenges. The biopsies received from lung cancer patients are often very small, thus containing only a few tumor cells. Then another problem, turnaround times have a great impact on prognosis of the patients. So the earlier the therapy can be started, the better the outcome. And furthermore, we face often quality restrictions due to fixation of the samples. For these reasons, we need an NGS panel dealing well with all these challenges when analyzing lung cancer samples. And now I'm going to explain why the OncoMine-Dx Express test panel is a potential candidate for our application. So one reason is the XCT panel is an amplicon-based panel with short amplicon size, around 150 base pairs, using molecular barcoding. This helps in reducing FFPE artifacts and even enables sequencing in cases of poor FFPE tissue sample quality. And furthermore, with this panel, we are able to analyze CF DNA samples. The panel requires only 10 nanogram DNA RNA input, thus dealing well with small sample size. DNA and RNA can be analyzed simultaneously, and sequencing on the GeneXus DX system enables fast sample processing with few hands-on time. In our lab, we were able to process 95% of the samples within 5 business days upon arrival, and 82% can be even processed within 3 business days. And we have a very low failure rate, which is indeed below 1%. But this slide shows an overview about the targets covered by the OncoMine-Dx Express test. The genes highlighted in red here are the genes which are important for lung cancer. In addition, we have the genes STK11 and KEAP1 now in this assay. And why is this important? So STK11 encodes the protein LKP1, which is a serine tyrosine kinase, being involved in the regulation of cell metabolism via AMP kinase signaling. And this gene acts as a tumor suppressor gene. Inactivating mutations in this gene occur in around 20% of non-small cell lung cancer. Mutation in this gene lead to a reduced response on PD-L1 based immunotherapy, especially in KRAS mutated non-small cell lung cancer. Thus, this is an important prognostic marker and often requested by the oncologists. The same is true for the gene KEAP1, which is a negative regulator of the NRF2 mediated antioxidant response. Similar to STK11, KEAP1 acts as a tumor suppressor gene, and inactivating mutations occur in approximately 20% of NSCLC. STK11 and KEAP1 are often co-mutated in NSCLC, and KEAP1 mutations may lead to a reduced response on PD-L1 based immunotherapy as well, especially in KRAS mutated NSCLC. Now I am coming to some examples in which we could receive utilizable results when analyzing very small lung biopsies. So on the picture on the left, you can see an hematoxylin eosin staining of an NSCLC sample. In this sample, we see only a few scattered tumor cells. DNA concentration was pretty low, but however, we could use 10 nanogram DNA, and in this case, we could detect the therapeutically relevant exon 20 insertion in the gene EGFR. So the next example shows a fine needle aspiration of a pleural carcinomatosis of a pulmonary adenocarcinoma. Here as well, only a few scattered tumor cells are visible. And what we found on RNA level, we found a fusion between the genes EML4 and ALK. This is a typical driver event in non-small cell lung cancer with a great therapeutical relevance. We could confirm this by ALK immunohistochemistry, and furthermore, the presence of ALK fusion event could be confirmed by the expression imbalance assay, which is part of the OncoMindX Express test. Okay. So now I'm coming to the next part of the presentation, which focuses on the analysis of cell-free DNA. So cell-free DNA is released into the plasma by several cell death mechanisms or even by active secretion. cfDNA may arise both from cancer cells, but also from non-tumor cells. And cfDNA released by tumor cells is called circulating tumor DNA, or abbreviated ctDNA. So why we should think about liquid biopsy? So liquid biopsy is an alternative approach if biopsy of a tumor is not possible or no adequate tissue sample is available. It is a minimal invasive method and allows monitoring of therapy response. And furthermore, it may represent not only a single tumor location, thus may overcoming potential tumor heterogeneity. So what are the challenges in analyzing liquid biopsy? So depending on the proportion of actual tumor DNA in the cell-free DNA sample, the variant allele frequency of potential variants may be pretty low. It's often below 5% or even below 1%. And for this reason, we need deep sequencing to detect these variants. cfDNA samples do not only consist of circulating tumor DNA, since non-tumor cells release cfDNA as well. And in addition, leukocyte lysis may release genomic DNA into the liquid biopsy sample, and this would lead to a wild-type bias of the sequencing analysis and thus to a reduction of sensitivity. And for these reasons, the pre-analytic workflow has a critical impact on the sample results, and this requires proper quality assessment of the respective samples. How can we do this? So one possibility to prove quality of a cfDNA sample is electrophoresis. So cfDNA shows typically a peak approximately around 166 base pairs. And this arises from mononucleosomal units of DNA, which derive mainly from apoptotic cells. And apoptotic cells are the main source of cfDNA. In contrast, genomic DNA would show a peak around 700 base pairs and larger. In this isolate, the purity of cfDNA was estimated to 98%. So this is example for a good sample. Now this slide shows an example for a cfDNA sample which is of poor quality. Here you can see a peak around 700 base pairs, and this resembles the potential contamination with genomic DNA, and in this case, purity of cfDNA was only around 74%. Okay, so this genomic DNA may originate from lysed leukocytes. I am going to explain now why this could be very critical when analyzing liquid biopsy samples. So the problem is leukocytes may harbor mutations due to clonal hematopoiesis, and clonal hematopoiesis of indeterminate potential, abbreviated with CHIP, is an event which may even occur in healthy individuals. And some of the genes affected by CHIP are genes that are covered by the OncoMIND-X Express test. For example, TP53, KRAS, BRAF, NRAS, and IDH1 or 2. And thus, if we find a mutation in these genes, it must not necessarily mean that this mutation is tumor associated. So we need to be pretty careful when interpreting liquid biopsy results, especially if we don't have any information about the mutational status of the tumor. To shed light into the dark in such cases, additional sequencing of leukocytes may be an option. Now I'd like to proceed with a case study. So a 70-year-old female patient was hospitalized on the intensive care unit due to severe dyspnea. There was suspicion of non-small cell lung cancer. However, a biopsy was not possible. The clinicians analyzed the sputum of the patient and they found evidence of malignant cells. And then they requested us to do liquid biopsy analysis in order to find a therapeutically relevant target. So first, we isolated cell-free total nucleic acids using 4 milliliter plasma. And then we determined concentration and performed quality assessment using electrophoresis. And the results are shown here. In this sample, purity was pretty good, 98% cfDNA, almost no contamination with genomic DNA. The plasma workflow of the OncoMIND-X panel, so we performed sequencing using the OncoMIND-X panel, covers all the relevant lung cancer genes, which are outlined in blue here. And with 30 nanogram cfDNA input, we can obtain limit of detections below 0.5%. This slide here gives an overview about the quality criteria of the respective sequencing analysis. We could achieve a mean depth of more than 50,000 reads and a median molecular depth of more than 2,000. And indeed, 91% of the amplicons show a limit of detection below 0.5% and 80% even around 0.1%, which is pretty good. And what we found, so we found a variant leading to the substitution of the amino acid leucine at position 858 to an arginine in the EGFR gene. This variant resembles a typical hotspot mutation, which is highly specific for non-small cell lung cancer. And based on these findings, the patient received a targeted therapy using an EGFR tyrosine kinase inhibitor. The patient responded very well to the therapy so that he was transferred from the ICU to the normal ward after 1 week, and a further week later, the patient could even be released home. So the patient responded pretty good to the therapy. So the ODxET workflow provides optimal results for our lung cancer patients due to a good target selection for non-small cell lung cancer, low turnaround times, good results even in case of small biopsies and poor sample quality. And furthermore, the liquid biopsy workflow shows good target selection for NSCLC as well, and is mainly used in the case of not biopsy-able tumors. So now I'd like to point out some limitations of the ODxET panel. The ODxET panel can only detect mutations if they are defined in the corresponding hotspot file. This means if variants are not contained in this file, they cannot be detected even if they are present in the respective BAM files. Since the ODxET assays and CE-IVD assay, there is no customization of the bioinformatic workflows possible. And I'd like to show you an example here. So in this case, expanded analysis using the integrative genome browser revealed a 16 base pair deletion in the splice acceptor site of the gene MET. And such mutations lead to MET exon 14 skipping, which is an important therapeutically relevant change in lung cancer. The presence of MET exon 14 skipping could be confirmed on RNA level, which is part of the ODxET assay. However, the variant on DNA level could not be called by the ODxET assay because it was not located in the hotspot file. However, expanded analysis in the IGV clearly shows that there is a mutation at this point affecting the MET, the splicing of MET exon 14. Now, one may argue there is detection on RNA. However, we need to know that detection RNA level is not always possible, for instance, due to quality reasons. Thus, there may be some risk of losing important information. So what we need to do in some cases, so we recommend reporting such cases to the bioinformatics teams of Thermo Fisher. And fortunately, there's continuous expansion, adaptation of the bioinformatic workflows through software updates. However, the colleagues need to know. So whenever you find a variant like this, you should report it to the team that they know and improve. To my opinion, it would make sense to even allow de novo calling for certain groups of variants, for example, MET exon 14 skipping, EGFR exon 19 deletions, KIT exon 11 deletions as well, to prevent some loss of information. The slide here summarizes the most important advantages and disadvantages of the ODxET panel. Indeed, in our applications, the advantages of the panel clearly outweigh the disadvantages, providing important factors for satisfied oncologists and pulmonologists. So one important point is that we get results from little material, which is often the case in lung cancer samples, and as well short turnaround times, which are pretty important because the earlier therapy can be started, the better. With this, I'm at the end. Thank you very much for your attention, and I'm open for questions. Thank you.